One of the key words of the brain, as we all know, is the word complexity. We study the endocannabinoid system, the receptor CB1, exactly as a tool to enter a little bit into this complexity of the brain. And this month we had this great chance to have two papers published on apparently different subjects, both of them involving CB1 receptor, which is able to regulate the functions of the brain really through different mechanisms and therefore give us some hints on the general way in which the brain works. On one story, we could show that CB1 receptors are expressed and present on brain astrocytes. And the most important thing that we found here is that it is this subpopulation of receptors that is responsible for the deleterious effects of THC on short-term memory, on working memory. This was made in collaboration with a group of Xia Zhang in Canada and with other people in the lab, and particularly with Mathilde Metna in my group. And we could show by deleting CB1 specifically in astrocytes that these animals became unresponsive to this specific effect on spatial working memory of THC. The other story that we published in these days is possibly even more intriguing for me, because as we know, CB1 receptors are G protein coupled receptors, and the classical way, the textbook way in which we think of them is that the receptor is sitting on the cell membrane and there exerts the functions of regulation of a cell. However, what we found together with Giovanni Benard and other people in the lab is that indeed CB1 is not only present at the membrane, but is also present on the mitochondria of the neurons. And this is a kind of new thing, because so far nobody could had even almost thought of the possibility that G protein coupled receptors are on mitochondria. And the interesting thing is that we found that this CB1, that we call it's still the same protein, but we call it MTCB1 to distinguish from the other population. This is able to regulate mitochondrial activity in neurons, and thereby possibly regulate also synaptic transmission and plasticity. This point of energy use of the brain is very important, and I'm very happy that the CB1, that is my subject, is involved in this, because we have to consider that, for example, the brain represents only 2% of the body weight, but it can consume up to 20% of the energy of the body. And therefore, this idea that how we think and we do things and how the brain works is strictly related to the use of energy is an old concept, but is becoming a new field of research that is becoming very interesting to understand how the brain works, both in physiology and in pathology. So I'm very happy that we had this chance to have these two papers published in this moment, but mostly because I think that these papers represent kind of way in which we can think that things that are thought to be almost impossible, when we study the brain, we realize that they are indeed possible, and we can find the way to show that they are very likely like that. I would like to thank all the people in the lab, particularly Mathilde Metna, as I said, Giovanni Benard, and Federico Massa for the work done in these two papers. And I thank you for your attention.